This study delves into the challenges of predicting traits from images lacking visible cues, particularly in the context of biomedical sciences. The authors focus on predicting MGMT methylation status from MRI images of glioma patients, which is crucial for treatment decisions. Despite numerous efforts, contemporary models exhibit suboptimal performance, and the underlying reasons for this limitation remain unclear. To address this issue, the authors conducted benchmarks of existing models using transfer learning, dissected their architectures by observing gradient flow across layers, and applied a feature selection strategy to enhance model interpretability. Their findings suggest that current models are unlearnable and may require new architectures to explore applications in the real world. This research aims to draw immediate attention and catalyze advancements in predictive modeling with non-visible cues. The study focuses on predicting MGMT status from MRI images, a critical task in glioblastoma diagnosis. Despite numerous efforts, contemporary models exhibit suboptimal performance, and the underlying reasons remain unclear. The authors aim to investigate this problem through a process-driven approach, identifying the necessity of indigenous tools in this domain. The dataset used is from the Brain Tumor Radiogenomic Classification Challenge, BRATS 2021, consisting of 585 glioblastoma patients' MRI scans, divided into unmethylated and methylated MGMT groups. The images are pre-processed and resampled, and all four imaging modalities, T1, T1CE, T2, and FLAIR, are utilized. The authors employed standard convolutional neural network (CNN) architectures, such as ResNet, DenseNet, and EfficientNet, as baseline models. ADAM and RMS prop optimizers, along with hinge and BCE logit loss functions, were used. The model's predictive efficacy was assessed using accuracy, AUROC, sensitivity, and specificity. Transfer learning was also explored, using ImageNet and MedNet weights for model weight initialization. Two approaches were employed, using previously trained models as feature extractors and fine-tuning the complete architecture with initial MedNet weights. To enhance predictive capability, an explainable approach was adopted. Pyradiomics was used to extract various features from MRI images, including first-order statistics, shape-based features, and texture features. The Mann-Whitney U-test was conducted to identify statistically significant features, which were then retained for further analysis. Recursive feature elimination using a random forest algorithm was employed to identify the most informative features, which were used to construct a predictive model. The study's comprehensive exploration involves benchmarking existing models, studying gradient flow across layers, and applying a feature selection strategy. The authors aim to demystify the complexity of MGMT status prediction, identifying the necessity of indigenous tools in this domain and discussing possible solutions to persistent saturation. The research focuses on developing novel architectures for MRI image analysis, leveraging hierarchical clustering to identify significant feature groups. An algorithm for recursive elimination of features is employed, using cross-validation to optimize feature selection. This process guides the development of novel architectures. The predictive performance of various CNN models, including DenseNet, ResNet, and EfficientNet, is evaluated across different MRI image types. Despite exhibiting suboptimal predictive accuracy, the model's performance varies across image modalities and folds, underscoring the challenges in this predictive task. The ResNet is chosen for its parameter efficiency and consistent performance. The T2 image type is selected for its ability to capture tumor characteristics. The study also explores the impact of error metrics and optimizers on predictive performance, showing that while improvements are observed in training, these gains do not translate to the testing dataset. The research highlights the limitations of training complex models on small datasets and the potential benefits of transfer learning approaches to improve model performance. Here is the combined summary. The authors investigate the effectiveness of various hyperparameter combinations, including loss functions and error metrics, on model accuracy. Figure 1 demonstrates the comparison of these combinations on both training and test datasets, highlighting the overlap of accuracy for hinge with RMS and ADAM. The paper then delves into the application of transfer learning approaches to improve model accuracy in predicting MGMT status from MRI images. Two types of transfer learning methods are employed: interdomain and intradomain. In the interdomain approach, ImageNet weights are used, whereas in the intradomain approach, MedNet weights are utilized. Initially, the ResNet10 model is employed as a feature extractor, where the model's weights are fixed, and only the fully connected layers are trained. However, this approach fails to significantly improve predictive performance, suggesting that the learned features do not effectively correlate with output labels. To address this limitation, the authors fine-tune the ResNet architecture with pre-initialized MedNet weights, training the complete network using the BRATS dataset. This approach yields noticeable enhancements in predictive accuracy on the training data, up to 70%, indicating that the model adapts its features more effectively to the task. However, the test dataset's performance remains unchanged over the first 20 epochs and later dips below 60%. This outcome raises concerns about overfitting and the model's ability to generalize its performance on test data. The authors argue that further investigation into model behavior is crucial, including analysis of parameters like sensitivity and specificity and the learning curve. Additionally, examining the gradient flow is essential to determine if the model is learning meaningful patterns. If the weights are consistently updated across epochs, eventually reaching a plateau, the model may have learned all it can from the data, indicating a generalization problem. Otherwise, it is a pattern recognizing problem. Model investigation for saturated accuracy reveals the relationship between a model's performance, measured by accuracy, and the number of training epochs. The learning curve in Figure 3 displays the fluctuation of sensitivity and specificity in both training and test datasets, showcasing complementary and erratic behavior in early epochs of the training dataset and entire epochs of the test dataset. This insight suggests that the observed lower accuracy of 60% is not due to learning behavior but rather random favor causing either lower sensitivity or specificity. Inspecting the gradient flow in training epochs, as shown in Figure 4, reveals that model weights remain unchanged despite training over multiple epochs, indicating the model is not effectively learning from the training data, leading to suboptimal performance and erratic predictions. Furthermore, certain model layers are not receiving new gradients during backpropagation, resulting in minimal weight updates, particularly prominent in the initial layer that captures major imaging features. The investigation highlights the need for models to learn effective behavior required for predictions. To analyze the relationship between complexity and model performance, the authors increase the complexity of ResNet by adding convolution layers in 3 sets, namely 10, 34, and 50. The results, presented in Table 2, show no clear dependence between performance in terms of accuracy and AUROC, with both low and high complexity models yielding accuracy between 5,260%. This suggests that model accuracy is independent of its complexity, leading to the conclusion that current models are unlearnable and fail to incorporate imaging features. Moreover, simple architectures could also be sufficient, and increasing model complexity may not be necessary. In the context of predicting MGMT methylation status in brain tumors, this research explores various CNN architectures and training methodologies to identify optimal models. Table 2 highlights the relationship between model complexity and predictive performance, revealing that ResNet10 and ResNet34 models demonstrate competitive accuracy and AUROC scores across different folds. The study suggests that these models could be effective in capturing subtle patterns in images, provided they are guided by specific domain knowledge to improve the current regime. To complement the CNNs, radiomics features were extracted to provide insights into image characteristics. Figure 5a illustrates statistically significant features across modalities, with FLAIR showing the highest number of significant features. The overlap of significant features among FLAIR, T1, and T1CE modalities is depicted in Figure 5b, indicating a strong correlation between these modalities. Feature selection using recursive elimination was applied to determine the best features for each image, resulting in varying numbers of selected features for each modality, Figure 5c. FLAIR selected 6 features and achieved the highest performance, suggesting its potential as a key modality for predicting MGMT methylation status. The clustering of similar features using a dendrogram, Figure 5D, reveals 3 clusters with 2 features each, further emphasizing the importance of FLAIR in this context. This study demonstrates the potential of combining CNNs with radiomics features to enhance predictive performance in identifying MGMT methylation status in brain tumors. The findings underscore the need for domain-specific knowledge to guide the algorithm and improve the current regime. The study delves into the complexities of predicting MGMT methylation status from MRI images, a task that is challenging due to the subtlety of non-visible cues. It highlights the limitations of applying techniques designed for capturing strong visible patterns to recognize minor variances not observed visually. The research emphasizes the need for an in-depth exploration to identify indigenous tools in this domain, as current solutions may not be tailored to address such problems effectively. The paper presents a case study on predicting MGMT methylation status, demonstrating that model performances remained unchanged despite adjustments and the exploration of transfer learning techniques. A detailed examination of the learning curve revealed poor learnability, indicating challenges in the training process. This suggests that it is not fair to use techniques designed for strong visible patterns to recognize subtle minor variances that are not observed visually. To enhance model architectures, the study proposes an explainable approach which provided insights into potential avenues for improvement. Adding influential radiomic features may enhance performance but could also introduce bias, necessitating further exploration. The current study aims to draw attention to the sensitivity of the problem, providing a possible direction towards a solution rather than a concrete solution itself. In future research, the authors plan to develop custom feature maps informed by the extracted knowledge of essential features, thereby addressing the underlying identified issues. The paper concludes that while the identified features are important, understanding their association is crucial. A dendrogram analysis showed 3 clusters with 2 features each, suggesting these groups can be used to derive custom feature maps. Implementing knowledge-driven feature maps can enhance the learning of convolution networks, but caution must be exercised to mitigate biases and ensure generalization ability across diverse datasets. The study also provides a comprehensive list of references, including works on radiogenomics, precision medicine for acute myeloid leukemia, and predicting 1p19q codeletion status from MRI images using deep learning in adult-type diffuse lower-grade gliomas. This research paper discusses the potential of using machine learning algorithms and MRI-based radiomics for non-invasive prediction of genetic mutations in various types of cancers and diseases. It highlights several studies that have demonstrated the efficacy of these approaches in identifying specific genetic markers, such as IDH mutation and H3K27M mutation, which are crucial for treatment planning and patient outcomes. The paper begins by discussing the importance of non-invasive diagnostics in personalized healthcare, citing clinical and translational science studies that emphasize the role of AI in this field. It then delves into various studies on the use of MRI-based radiomics and deep learning algorithms to predict genetic mutations in different types of cancers, including rectal cancer, breast cancer, and brainstem gliomas. For instance, a study by Meng et al. 2019 utilized MRI-based deep learning radiomics to predict the disease-free survival of patients with breast cancer. Similarly, Kandamerli et al. 2021 employed a machine learning-based multiparametric MRI radiomics model to predict H3K27M mutation in midline gliomas. These studies demonstrate the potential of these approaches in improving diagnostic accuracy and treatment planning. The paper also touches upon the role of MGMT promoter methylation in predicting response to radiotherapy and its prognostic value in the absence of adjuvant alkylating chemotherapy for glioblastoma, as discussed in Rivera et al. 2010. Moreover, Capuazzo et al. 2022 proposed a multimodal knowledge-based deep learning approach for MGMT promoter methylation identification, further underscoring the versatility of machine learning algorithms in this context. In conclusion, the paper presents a comprehensive overview of the current state-of-the-art in using machine learning algorithms and MRI-based radiomics for non-invasive prediction of genetic mutations in various diseases. It highlights the potential of these approaches in improving diagnostic accuracy and treatment planning, while also emphasizing the need for further research to fully realize their potential. The research presented on pages 17 and 18 focuses on molecular subtype prediction and radiogenomic classification for management promoter methylation status using multiomics fused feature spaces. It discusses various methodologies, including UNet-based segmentation, adversarial learning, and attentive multimodal CNNs for brain tumor radiogenomic classification. The studies leverage MRI scans to predict management promoter methylation, with some exploring the potential of transfer learning and deep learning models for this purpose. The results highlight the significance of radiogenomics in least invasive diagnosis. Key findings include the effectiveness of multimodal approaches and the potential of vision graph neural networks for management promoter methylation prediction. The limitations and future work discussed emphasize the need for further investigation into the robustness of these methods across different datasets and the integration of additional modalities for improved accuracy. The combined summary of pages 19 and 20 focuses on various aspects of machine learning, particularly in the fields of computer vision, transfer learning, and image processing. It highlights the significance of deep residual learning for image recognition, densely connected convolutional networks, and efficient model scaling for convolutional neural networks. The summary also emphasizes the importance of transfer learning in different domains, including medical image analysis and speech disorder classification. Additionally, it touches on multi-class 3D object detection in baggage security screening and machine learning-based compressive strength prediction for concrete. Throughout, the summary maintains a technical and professional tone, ensuring the flow of ideas mirrors the structure of the original pages.